Hi. Thank you again for the organizers. Such a lovely meeting. And I'm gonna talk to you about today about the interplay between fertility and aging, and how these two phenomena that are critical are intertwined, and can we say something from one phenomena on the other? So, okay, there's for the young guys, I think that after we're all in the middle of the second day, so you realize that we all age, right? We cannot escape from that. So here's a graph that shows what happened to the mean life expectancy in the last 2 years, and you can see that we're doing good, right? Besides the two World War wars dips over there, the mean lifespan is increasing. But if you look on the right-hand side, where you can see the maximal lifespan, you can see that we are not really changing it. Okay, so it's about 120 years, and this is basically our main challenge, because extension in mean lifespan can be attributed to hygiene and antibiotics and diseases, and you know, as far as it goes to maximal lifespan, really flat, and that it bring up the question: so why is it that? Is that a limit? What can we do with that limit? And I want to. So we all age. That's a fact. And the question maybe is why we age, right? And when you think about it, why we age, you can think about two limiting answers, right? So the first answer is, okay, things just break down, you know, wear and tear, entropy is the queen, you know, natural selection couldn't find ways to deal with the trade-off that is associated with repair and growth in complex systems like human beings, because we know that there are systems that can rejuvenate, biological systems that can rejuvenate forever, right? So, and I guess some of you know all these theories, you know, disposable soma, all kind of trade-offs, and that's why we age in complex systems. The other way to think about it, the other limit, is that actually aging is not the failure of natural selection. It's not a bug, it's a feature, okay? So aging is actually a selected trait. So if aging is a selected trait, the lifespan is actually something that has been evolved to be the way it is. And the tension between these two hypotheses is not just, you know, a philosophical debate over lunch. It actually really determines the way we cope with how to deal with aging. So should we deal humans like cars? Okay, so we just repair, you know, or should we try to understand the underlying biological programmed aging, if you'd like, in order to really extend the maximal lifespan? So how can we get a glimpse, some experimental glimpse on the tension between these two hypotheses? And that's where fertility and reproduction senescence comes into picture. So what you can see here in this graph is the on the y-axis is the reproduction senescence age, where the organism stops to be fertile, and on the x-axis is the maximal lifespan. This is for mammals, and you can see that the longer you live, the more fertile you are, but this line is not linear, right? You can see that there is this bending that we are not really quite understand yet. All the models for that all of them have a lot of limitations. And there is a gap. Okay, so the longer you live as a mammal, the more time you spend as a non-fertile organism, right? And that's again raised the question of, okay, so if there's program aging, let's say that this is something that there is, so why are you alive for so long and not being fertile, right? So aging is different, the aging process is different. Is it accelerated? Does it start when you are stopped to be fertile? So the transition between fertile and non-fertile organism and how aging is progress along with it could be insightful to the question we just raised. And that's what we try to do in my lab, among other stuff. And what I will focus today is on the question of how immune fidelity changes with age with respect to reproduction senescence. And that's a wonderful, it was done by a wonderful MD PhD student, Tal Ben Yaakov. So this is his work. He did all the heavy lifting here. So the main idea is, let's say that we think about immune fidelity, some function, as a function of age. So we have a lot of work, the upcoming these years about inflammaging, right? That's a major phenomena that happens where in older organism there's an increase in inflammatory markers. The immune system becomes more inflammatory, become dysregulated, and that could lead to chronic diseases in all the organism and all people. But does inflammaging is again just a wear and tear that happens, as we age, we deteriorate, and the immune system becomes more and more and more bad, or whether the inflammaging process or immune fidelity in general actually start to deteriorate with respect or with correlation with reproduction senescence. And what Tal was doing, we have a great or a model for fertile non-fertile transition, which is the mammalian female, right? So we have menopause in women, estrapause in mice. So we have a very sharp transition between fertile and non-fertile. So what Tal did is to focus on the ovarian immune system. So the ovary is really fascinating in this respect because immune system plays a crucial role in many, many processes regarding to oocyte maturation in the ovary. And on the top of that, because of the cycles, the ovary is a very inflammatory organ. Okay, so if you think about it, if there is inflammaging, each cycle is actually a very inflammatory process. So if there's a place that you don't want to have inflammaging happens, is while there is cycles in the ovary. So Tal actually asked himself, okay, if we go to the literature, if you look on very, very old mice, it's like 2 years old, it's like 100 years old in human scale. Is there inflammaging? There is. So actually every old mammals we measured, if you go, 2 days before they die, there is some type of inflammaging. But does the aged fertile female has also inflammation processes? Okay, so what is the changes in the immune milieu along the entire aging axis, not just at the end? Okay, so what we did here, we took samples from the ovary in mice in different age groups, and we just analyzed the hell out of them. Okay, whatever we can measure, we measure. So the first result is that the immune milieu in the ovary is very complex. Okay, we see a lot of cells, and we see a lot of non-canonical cells, if you'd like. For those of you who are more on the immunology side of things, I would just say that we have very unique populations like ILC1 that we don't really see in the blood or in the liver or in the spleen. And we have a population of double negative, CD8 negative, CD4 negative cells, types of T cells, which are not gamma delta, which are really unique to the ovary. So that's very interesting if you're immunologist, okay? This was the first characterization, the first single cell characterization of the immune milieu, but we're interested in what happens with age. And so what you can see here is the changes in frequency of different immune cells in the ovary. And the main changes, I will not get into all the details, is that we have a decrease in CD3 lymphocytes, in macrophages, and increase in CD3 positive lymphocytes. So we have an increase towards adaptive immunity, if you'd like. Okay. And we validated these results also on the protein level, not just the mRNA level, and we saw something similar. So that's what you can see here is the summary of both transcriptome analysis and protein analysis that shows that both of them are consistent, and overall there's a shift towards adaptive immunity in the ovary. Now, how does that relate to inflammaging? Well, it's not, okay? Because if inflammation is not transition towards adaptive immunity. So we wanted to investigate it more, and so far I showed you results about the fractions of cells, the populations of immune cells, what we can say about the actual biology of the cells. So you can see here analysis that shows for each cell group, for each immune cell groups, what is changing in their biological process. And we see a lot of things that we expect to see. You know, when my youngest daughter asked me, what do you do in the lab? I say, okay, we study aging. And she says, but doesn't everything just go bad, right? And it just all go bad, right? And so you can see a lot of these type of figures, you know, it goes bad, you know. Transcription goes down, cellular metabolism goes down, right? So, okay, this is not really, it's interesting, but it's not insightful. So what we wanted to see is what happens to the actual interaction between immune cells. So we looked on the dynamics of chemokines and chemokines and cytokines. So my students call them together chemokines. They combine the words to save saying both of them together. So I did it also. These are small molecules that immune cells are secreting in order to regulate each other. And what you can see here in this table is the effect of age on the transcriptome level of these chemokines and cytokines. And you can see that in general, it's all red, right? So you could say, okay, you just told us that on aging, everything just goes down, so maybe it's a global effect, but it's not. Okay, so that's normalized already by the global decrease of the changes due to aging. And interestingly, what you can see here, that we don't see any increase in inflammatory markers. Okay, so I remind you, old here is the equivalent of 55 in humans. So they are still before menopause. Okay, so no inflammaging, there's no inflammatory markers that goes up. Actually, all the inflammatory markers, there's a bunch of names here. They all go down. Okay, so that was very interesting, and we validated also experimentally, because again, this is just mRNA results. And indeed, most of chemokines and cytokines that are inflammatory go down. Okay, there's no inflammaging here at all. The second thing that we saw that was very interesting is one of the tasks of the immune cells is also to recognize senescent cells. So senescent cells are cells that undergo growth arrest due to damage or aging, and once they become senescent, they start to secrete some small molecules, which are then being recognized by part of the immune cells to cope with them. Okay, and what we found out, interestingly, that in macrophages and neutrophils, there's an increase with age in the receptors for these molecules. So with age, the macrophages and the neutrophils, which have the job of deal with senescent cells, are actually increasing their receptors. They're becoming more sensitive to senescent cells. So overall, if I summarize what we saw here, is that while the mice at least ages and still fertile, not only that there is no inflammaging, it looks like the immune system is doing quite a good job to deal with the two main challenges that arises with aging, again, in the fertile female. The first one is the repeated cycles. Okay, so there's inflammation and inflammation and inflammation. So what happens is there's a reduction in the inflammatory state to cope with these repeated cycles. And the second thing is the increase in senescent cells. So that summarizes here. So what we think has happened is that we know that when the organism is very, very old, there's inflammation. But I'm talking about females now. But again, this is 1 year into non-fertile condition. At least in the fertile aging trajectory, there's no inflammation, and it looks like the immune system is doing really well. It's able to cope with the task. So inflammation is not an inevitable process, okay? It's something that is probably controlled, at least at mice. And the overall result, I think, raises an interesting point that if we want to study aging, it's a good idea, at least we think so, to start not only at the end. Okay, you really want to monitor the aging process across the entire lifespan of the organism, because this example absolutely demonstrates that the aging phenotypes that we like to think about are not unidirectional, non-reversible states. And I want to thank you, and if you have any questions, I'm happy to take them.
Okay. We know that when we're born almost like half-baked. Essentially, we're not able to take care of ourselves, and we need our mom. And we need our mother to take care of us, and essentially through her milk we get immune support. Now, after menopause and aging, you still have grandma around. So do you think there is a role for the immune system essentially to be a part of maintaining social aspects of our being as a society, not necessarily as individuals?
Yeah, so that's of
maybe
course,
a little philosophical.
Yeah, that's a great question. So in the evolutionary kind of, this is called the grandmother paradox, because it's, again, why do we need the grandmothers, right? They're not fertile. Why aren't they like just drop dead, right? As it happens in other organisms, right? So one of the ideas is a type of a kin selection kind of mechanism, for those of you who are more familiar with evolutionary jargon, which actually means that it helps its progenies. So that's a very compelling idea. The problem is that it doesn't really stick when you do the mathematical modeling. I would say that it's still an open question. The grandmother paradox, I think, is still a very interesting question. Yeah.
You were talking about fertility and the relationship for longevity, but what about the actualization of descendants, like having children or not having children? Could it have also some epigenetic impact?
Yeah, so we absolutely know that the act of having a progeny at different ages modulate your aging process, right? So if you have, at least in mammals, I'm talking about mammals now, only mammals. So if you have, okay, this is something that is, there's debate on it, whether if you have kids at late age, whether you live longer. Does it increase your lifespan? Because there's a lot of social reason for that and whatever, and a lot of biases. But in general, we can see it also in lower mammals. What is the underlying mechanism that gives you this boost is not really known. There's probably some epigenetic thing in it, like in any other thing, there's some epigenetic change. But I don't think we can say for sure what is the mechanism that makes you live longer if you have kids later in life.
Please.
Any relationship between estrogen progesterone ratios, prolactin levels with your
Yeah, so that's a very, very, very interesting question, and the answer is absolutely yes. So all the hormone landscape really modulates a lot of the processes, also immune processes. Here in this work, what's interesting that there's almost no change in the hormone levels because they're still fertile. Okay, the major hormone changes happens after menopause or estropause in this case.
But
so it could be.
But cycling happens normally. You don't have. There's a cycle during the month. The hormones change.
Okay, so you're talking about the changes through the cycle. That's also a very good question. I haven't shown the slide, but the decrease in the immune changes is independent of the cycle. Absolutely independent. Our main hypothesis was actually that it's going to be cycle dependent. We expected that the changes would depend on the cycle, but they are not.
Check prolactin?
Yes.
Okay.
Hey, would you attribute the fact that if you hold the reproduction process, it's similar to what happens when you correlate it to fasting? So in a fasted state, your body knows that it needs to repair itself and that it's basically slowing down before the next feeding phase. And so
I'm not sure I understood the question.
So when you hold the reproduction system, right? You don't sort of reproduce quickly enough, right? Yeah. Your body might do the same thing that happens when you're fasting. So
in general, modulating the reproduction state. And I hope that's what you refer to, if I can make it fertile sooner or later.
Yeah, I'm just looking to basically
sorry, I just couldn't understand what you're saying. So sorry.
I'm just looking to find out if there's like similar mechanisms that are taking place as you know, when you're fasting, your body repairs itself. Yeah, when you fast in a fasted state. So your body.
Okay, now I got it. Yeah, I understand.
mTOR,
Yeah. So.
Involved. Sorry to interrupt.
Yeah, I got it.
I just want to understand if it's a similar process that's taking place when you don't reproduce early in life. Your body knows that it needs to wait longer, so it somehow extends your lifespan because it knows that you need to accomplish that reproduction mission later in life, so it prolongs your lifespan. I want to know if it's similar to what happens when you're fasting, so your body knows that it needs to slow its metabolism down, which also prolongs your lifespan to some degree.
Yeah, so let me just recap. So we know that fasting, along with other, it's very in trend these days, right? So we introduce some stress, fasting, some oxygen stress, some type of stresses, and this, gives the body some kind, the body is now reacting to this stress, and there's some rejuvenation processes that happens, and whether so there's no clear evidence. There is something interesting about pregnancies, okay, in that respect, because there are some initial results that if you have that, because a pregnancy is type of a stress, right? And we know that on some diseases. So that's a long answer for that. The question is that if you get a later pregnancy, okay, so you delay the first pregnancy, then you have some probability of not having a longer lifespan, but age-related diseases are delayed, like Alzheimer's, for example.
Thank you very much. I think we'll continue the discussion over the break, and we will continue with the Technion estafette with Shai Shannon.